Welcome back for another deep dive. Today, we're going microscopic. We're talking leukocyte migration.
Oh, cool.
Yeah. So those white blood cells, the immune warriors, they don't just sit around. They're constantly moving.
Right, right.
It's like super organized, going everywhere they need to, keeping us healthy. Our source material today is chapter 3 from this immunology text.
Okay.
It's all about leukocyte circulation and migration. So, like, how do they get around? Where do they go?
Yeah, it's amazing how they know where to be, right?
Exactly. That's what
Yeah.
we're diving into. So maybe we could start with the basics, like the main functions of this whole migration thing.
Sure. Well, first off, you got to get those myeloid cells, where the
Yeah.
action is.
Myeloid cells.
Yeah, like your neutrophils and monocytes.
Yeah, the first responders.
Exactly. They rush to infection sites, contain the damage, like a rapid response team.
So anytime there's inflammation, a cut, a cough, anything like that.
They're on it, drawn by signals released during that whole process.
Wow, that's so cool. So that's one function. What else?
Then there's guiding the lymphocytes.
Lymphocytes. You mean the T and B cells?
Yep. They need to get to the lymphoid organs. Like, think of it as boot camp for your immune cell.
Ah, so that's where they learn to fight specific threats.
Exactly.
Okay.
Places like lymph nodes, the spleen, that's where naive lymphocytes, the ones that haven't encountered anything yet, kind of go to school.
They're learning, training, becoming those specialized fighters.
Mhm.
Okay, so we've got the myeloid cells on the front lines, lymphocytes in boot camp. What's the third function then?
Sending out those trained lymphocytes.
So they graduate and go
Oh,
to...
wherever the battle is, that's how you get a targeted systemic defense.
Wow, so it's like a full-blown military operation, different units, strategic deployment. But how do they know where to go? Is there like cellular GPS?
It really is like that. And you're right, there's a whole system of signals and cues.
Okay, I'm all ears.
So, two key players, adhesion molecules and chemokines. Think of them as the roads and the traffic signals.
Ooh, interesting analogy. So adhesion molecules first, right? What are those exactly?
They're like the cellular glue.
Glue.
Yeah, they help leukocytes interact with blood vessel walls so they can get out into the tissues.
So they're on both the leukocytes and the endothelial cells lining the vessels.
Exactly. It's like a little handshake. And there are different types, like selectins.
Selectins.
They handle those first interactions, kind of like a gentle rolling along the blood vessel wall.
Like a patrol, checking things out.
Exactly. And there's different selectins, too. P-selectin, it's stored and ready to go, super fast response.
And then,
E-selectin, that one's made when needed, takes a bit longer.
gotcha. So like a rapid response team and then the specialists. But once a leukocyte finds trouble, how does it go from rolling to actually stopping and getting out?
Integrins, they're the strong grip.
Ah, so selectins are the handshake, integrins are the, like, grabbing on tight.
Perfect. But knowing where to grab on, that's where chemokines come in.
Chemokines, right. Those were the signals, the traffic lights. How do they work?
Imagine you're walking and you smell freshly baked pie.
Mom.
The closer you get, the stronger the smell, leading you right to it.
So the cells are following a scent trail.
Exactly. But different chemokines attract different cells. It's very specific.
So neutrophils might follow one scent, monocytes another.
Precisely. Each cell type has its own guiding chemokine. It's amazing. Right?
Totally. But wait, there's all these different types of cells, different functions, so their migration patterns must be different too, right?
Absolutely. Like neutrophils, they're the SWAT team. Super fast response, drawn by a chemokine called CXCL8.
CXCL8, got it. So what about those monocytes, the cleanup crew?
They're attracted by CCL2. They arrive a bit later, focus on cleaning up and repairing.
Longer lifespan, they can even multiply in the tissues. Right?
Yep. So it's not just fighting, it's the whole recovery process. Now, lymphocytes, they're a whole other story.
Yeah, the ones that go to boot camp. How do they get around?
Well, naive T cells, they're constantly moving, checking out those lymphoid organs.
Always on patrol.
Right. And they enter through these special blood vessels, high endothelial venules.
Like specialized entry points.
Exactly. And it's guided by, again, adhesion molecules and chemokines like L-selectin and this receptor called CCR7.
Wow. It's like they're playing hide and seek with their specific antigen. What happens if they find it? Do they stay in the lymph node forever?
Not at all. But I think maybe we should take a break here, come back, and delve into what happens after a T cell finds its match.
Ooh, cliffhanger. Okay, sounds good. We'll be right back after a short break. Okay, so we left off with those T cells on patrol in the lymph nodes. They're searching for their antigen match. But what about B cells? Do they just wander around too?
Well, kinda, but B cells, they have different hangout spots.
Hangout spots.
Yeah. They're drawn to the B cell follicles, like little marketplaces within those lymphoid organs.
Ah, so it's like different training grounds for different units.
Exactly. B cells meet their antigens there, get activated.
And that's guided by what, adhesion molecules and chemokines again?
You got it. One key player for B cells is a receptor called CXCR5.
CXCR5. Another one for the list.
It leads them right to those follicles where they can interact with antigens presented by other cells. It's all about connections.
Fascinating. Okay, so say a B cell finds its match in a follicle. What happens then? Does it stay there like those activated T cells?
Some do. They become plasma cells, start making antibodies right there in the follicle.
Antibody factories on site.
Exactly. But others, they're called plasmablasts.
Plasmablasts. Cool name.
They're the travelers.
Travelers. Where do they go?
A couple of favorite destinations. Some head for the bone marrow.
The bone marrow.
Yeah. They mature into long-lived plasma cells there, so you've got a constant antibody supply.
Like a backup reserve.
Exactly. Others, they prefer the front lines, mucosal tissues, lining the gut, respiratory tract, places like that.
Ah, so they're ready to intercept invaders right where they try to enter. Makes sense.
Right. And it's not just location. They actually produce different types of antibodies, each specialized for their environment.
Like what, different weapons for different battlefields?
Exactly. IgA antibodies, for example, they're great for neutralizing pathogens in mucosal areas.
Wow, so that's all strategically organized, like a multi-layered defense system.
Absolutely. But we've kind of glossed over something important. How do both T and B cells know when to leave the lymph node?
Now that you mention it, yeah. They don't just stay forever, right? There's got to be a signal.
There is, and it's super cool. It's a molecule called sphingosine 1-phosphate, S1P for short.
S1P. Okay, what's that all about?
It's like a homing beacon. Draws lymphocytes out of the lymph nodes and back into circulation.
Back into circulation, so they're constantly going back and forth.
Exactly. It's all about that concentration gradient. There's more S1P in the blood and lymph than in tissues.
So the cells are basically following a trail of this S1P.
Yep, and they have this receptor, S1PR1.
So when a naive lymphocyte enters a lymph node, S1P is low, so it hangs out, explores.
Right, and if it doesn't find its antigen match after a while, S1PR1 expression goes up.
Like, okay, time to move on.
Exactly, and the cell follows that S1P gradient out of the node.
So it's like a built-in timer, giving them a chance to find their match, or else back to the road. But what about when a lymphocyte does find its match? What happens to S1P then?
That's the really clever part. When a lymphocyte is activated, S1PR1 expression actually gets suppressed.
Pressed.
Yeah. So the cell can't see that S1P exit signal anymore. It stays in the lymph node, has time to multiply, specialize.
Ah, so they get their training, build up their forces before being redeployed.
Exactly. And once that's all done, S1PR1 expression comes back. They follow the S1P trail out.
Wow, so this S1P is like a master conductor, regulating the whole process. But I imagine, like anything complex, things can go wrong, right?
Of course. And that's when you see just how crucial this whole migration system is. Any disruption can cause big problems.
Like what kinds of problems?
Well, there are these rare genetic disorders, leukocyte adhesion deficiencies, or LADs. They really highlight the importance of those adhesion molecules we talked about.
LEDs. Okay, what happens in those?
Basically, the adhesion molecules are faulty, or the signals that control them are messed up.
So the cells can't move properly.
Exactly. Like in LAD1, there's a problem with a protein called CD18. It's part of several integrins.
Ah, so without those functioning integrins, no strong grip, no exiting the blood vessels.
Exactly. It's like trying to climb a wall with broken gear. And in LAD2, the problem is with those carbohydrate ligands that bind to selectins.
So no rolling interaction, no slowing down to check things out.
Right. And there's LAD3 too, which messes up the signaling between chemokine receptors and integrins. So even if they can roll, they can't get that signal to grab on tight.
Wow. So each type of LAD affects a different step in the process.
Exactly. And the consequences are pretty serious, unfortunately. Recurrent infections, delayed wound healing, all sorts of problems.
Because the immune cells just can't get where they need to be.
Exactly. It really shows how important this whole system is, how much we rely on it. It's not just some abstract concept. It's crucial for our health.
Absolutely. And it makes you think, if we can understand these processes so well, maybe we can also manipulate them, right?
That's a great point. And that leads us to a really interesting question.
Oh, what's that?
Well, we've talked about how important leukocyte migration is for a healthy immune system, but what about situations where we might want to, I don't know, limit or redirect that movement.
Ooh, interesting. Like when?
Well, think about autoimmune diseases. The immune system is attacking your own body. Could we somehow stop those cells from getting to those healthy tissues?
Oh, yeah. That's a big question. And maybe a topic for another deep dive.
Absolutely. There's always more to explore. But for now, I think we've covered a lot of ground.
We have, from those first steps of rolling along a blood vessel to the complexities of S1P and those LAD disorders. It's amazing how much we've learned.
And remember, this is all happening inside us right now. All these cells moving, communicating, protecting us. It's pretty incredible.
It really is.
Yeah.
But you're right, there's always more to discover. So, to our listeners, keep those questions coming. Who knows what other mysteries will unravel in the future?
And until next time, keep exploring that fascinating world within.
That idea of manipulating leukocyte migration, like to treat autoimmune diseases, it's kind of blowing my mind.
It really is a fascinating area, right? The potential is huge.
Imagine redirecting those immune cells, stopping them from attacking healthy tissue.
Exactly. And that's just one example. Think about vaccines, cancer immunotherapy, wound healing. If we could really control this movement, who knows what we could achieve?
It's like we're learning this whole new language, the language of the immune system.
Right. But it's complex. There's so much more to it than just adhesion molecules and chemokines.
Oh yeah, we just focus on those, but you mentioned other factors too, like what?
Well, think about the environment those cells are moving through. The extracellular matrix, all those surrounding structures, even physical forces, they all play a role.
Wow, so it's like the signals are important, but the whole landscape matters too.
Exactly. And we're still figuring all this out, It's an active area of research. For example, we've recently discovered these specialized memory T cells.
Specialized memory T cells.
Yeah, they actually live in specific tissues long term, kind of like a local defense force ready to respond if that same threat comes back.
Oh wow, so like each organ has its own little security team. That's amazing.
It is, and we're still learning how they work, how they're maintained, how they could be used to boost immunity. It's all very exciting.
It makes you realize just how much is going on inside us all the time. This whole world of cells moving, communicating, protecting us. It's mind-blowing.
It really is. It shows how amazing our bodies are, how they adapt, how resilient they are. I mean, think about it. Fighting off infections, repairing injuries, it's all happening constantly.
And we barely even notice. It makes you appreciate the power of scientific research too, right?
Yeah.
The more we learn, the more we realize how much we don't know.
Exactly. And who knows what future discoveries will bring? Maybe new ways to make vaccines work better, to develop therapies that target those misguided immune cells in autoimmune diseases.
Yeah, or even engineering immune cells with like super migration powers to fight cancer.
Exactly. It's all about understanding this intricate dance, so we can maybe learn to lead it someday.
Well, I think it's safe to say this deep dive has opened my eyes to a whole new level of appreciation for the immune system.
Me too. It's a reminder that even in the face of all these complex challenges, our bodies have these incredible mechanisms to keep us going.
And it all starts with something as seemingly simple as cells moving around. It's just amazing how much complexity and precision there is in that movement.
Absolutely. We've covered a lot today, but it's really just a glimpse into this whole fascinating world within us.
And the story's still being written, right? Every new discovery adds another layer to our understanding.
To our listeners out there, remember, keep exploring, keep asking questions, keep being curious. There's always more to learn about this incredible world inside us.
That's a perfect note to end on. Thanks for joining us in this deep dive, everyone. Until next time.